Dr. Stanley Jordan is very, very special to me. I've known him since I was a kid. He was a pediatric nephrologist when I was younger, and I always connected with him because I just felt his caring level was off the chart. And then fast forward, he has become the guru in understanding desensitization with people with high antibodies. I have a lot of presentations on our website about it. If you have high antibodies and anybody has ever told you you can't get a kidney because you have antibodies, that's not always true. It depends on the strength of the antibody, and we're learning more every day. And as a result of Dr. Jordan's amazing work, I've been transplanted 11 years and my creatinine is 0.7. So that's why I say he walks on water. And you just got to stay alive till the next miracle happens.
Okay.
So Dr. Jordan, I know now he's all, you deserve every accolade you get.
Oh, thank you.
And he today is going to talk about some innovation that's happening to help deceased donor kidneys be able to be used. So I'm going to turn it over to Dr. Stanley Jordan, who is the head of kidney transplant at Cedars-Sinai Hospital. Please welcome Stanley Jordan.
Well, thank you so much, Lori. And I really am looking forward to talking to you today about what I see as some of the newer areas of innovation, and try to focus mostly on the things that we're doing at Cedars. And I'm very excited about all of that. So the things that I want to, let me share my screen and see if I can pull up the presentation here. Okay, there we got a lot of stuff up there. Can you see it?
You can see your desktop.
There we go. Okay, here we go. Let me just do this. How's that? Is that okay?
Yes, that's good.
Okay, so it's going to be looking at emerging therapies for kidney transplant patients. And so we'll go through this in the next 15 minutes or so, and then hopefully I have some time for to answer some questions if you have any. We're going to focus today on new developments in transplant medicine that will likely, and I think dramatically, improve access and longevity of kidney transplants while limiting the current burden of immunosuppression. And this is something that's very important to talk about because, you know, our current transplant medicines are now over 30 years old, and we do need to move forward in this area. So we'll talk about as Lori mentioned, the new developments in desensitization and treatment of antibody rejection, which is a big problem for us. Looking towards more sustainable and less toxic transplant regimens, and then harnessing the body's own regulatory mechanisms to reduce inflammation, and then preventing ischemic or low oxygen injury to kidney transplants, and those implications for long-term graft survival. And we'll talk about each of these things today. Now, if we go back, way back, and I was at the 50-year celebration of this New England Journal paper in 2019, excuse me. And this is Dr. Terasaki, whom all of you know is a real famous immunologist in transplantation. And he was the first to show the importance of antibodies. At that time, they didn't know they were HLA antibodies, but they were antibodies that bound to the kidney and caused kidney loss. And he could use a person's lymphocytes as a surrogate to detect those before they damaged the kidney. And this led to the cross-match test. And this was a major advance in preventing kidney injury. However, the biggest problem is that it sort of marginalizes, as Lori said, you know, people to being on dialysis because there was no treatment to get rid of these antibodies. And so people tended just to stay on dialysis and not have an opportunity for transplantation. So I became interested in this many years later, and my focus has been on looking at ways we could get around this or develop new therapies to help prevent this. And as Lori mentioned, we've had success with a large number of therapies, including immune globulin and rituximab and plasmapheresis, but we want to move forward from that. And this is a picture 25 years ago when I was in India with these doctors, and you can see this patient here in the middle with his wife. They had traveled from Nepal to Delhi for kidney transplant. And this gentleman had what now we didn't understand at that time, but what now we would see as antibody-mediated rejection of his kidney. He had a positive cross-match after transplant, and it was destroying his kidney. At that time, we had no therapies except for steroids, and maybe some immune globulin was just coming around. So the journey from this place to where we are today, I think, is what I want to talk about. So if we look at kidney transplant survival over time, what we've found is that kidney transplant survival has never really improved, even in the modern day immunosuppression. After first 1 to 2 years, it goes back to where it was 20, 30 years ago. And people thought, well, that's because people take cyclosporin or Prograf, they have recurrence of diabetes, that sort of thing. But what was found out in this paper from France, and also this paper from US, is that most of the kidneys that are lost in the United States, probably in the world, are lost to what we call antibody rejection. So those antibodies that Dr. Terasaki detected many years ago do have importance post-transplant because they end up destroying most of the grafts. There's 5,000 transplants lost each year in the United States. All vintages, of course, but almost half of them are from this antibody rejection. It's become a major problem worldwide now to try to figure out therapies for this. And we were involved with one. Fortunately, had the opportunity to work with a company from Sweden. This paper we published in 2017, and we published more since, with an enzyme that comes from the bacteria Streptococcal pyogenes. And the bacteria is tormented by human antibodies, so it had to figure out a way to get around that. And so it made this enzyme called ITIS, and it would cleave human immune globulin in the half parts, so it wouldn't. So this prevented the antibodies from killing the bacteria, and it could flourish and cause death or really terrible disease in patients. Now you say, why in the heck would you want to work with something like that? Well, what we found is that this antibody, or excuse me, this enzyme, which cleaves the antibodies, is very useful in getting rid of HLA antibodies. All these red bars show, you have like 97 bars here of antibodies as a wall to prevent transplantation. But if you treat with this enzyme, you can see they go away. There's the little blue dots down here. And down on the bottom where we have what we call complement activating antibodies, the real bad ones, they're gone away in an hour. So this was a revelation, and we reported this in New England Journal back in 2017. And we've had several articles since then, but this was I was so proud of this because the work we did at Cedars and then the groups in Sweden as well allowed this drug to be approved, first drug approved since 2010 for any condition in transplant for desensitization, which is such a great thing. So it is now out there and is available in Europe. We're working on a study in the US to have it approved here with the FDA. So the other target is what we call interleukin-6. This is a cytokine that is we found is very important in it causing inflammation in the kidney and causing rejection. And we started doing work with this back in this paper we published in 2017. We started working around 2011. You can see this is a 6-year study showing that if we treated people who had this chronic antibody rejection that causes 50% of the graft losses, that we could really stabilize kidney function and improve graft survival compared to standard of care or SOC. And more importantly, on the right side, there was a life-saving benefit to this drug, because people who lost their kidneys from rejection tended to do very poorly on dialysis and had a higher risk for dying. So we see this as a very, very important therapy. And so I won't spend too much time on this, but we've got now a new drug. It's very similar to tocilizumab, but more powerful. That is in evaluation in a worldwide study for treating antibody rejection. And this just shows you some data we had for early studies at Cedars that if you look below here, all those lines above, I won't bother you with, but this is like 24 months before the patients entered our study, you can see their what we call GFR, which is their glomerular filtration rate, or how their kidneys working. Higher the better there. And at the time they entered the study, it was dropped very significantly. But after 2 years of treatment, it was really stabilized. So we feel like we can stabilize these kidneys. And this is what the study will be looking at for a labeling. And here in desensitization, it's one of the best things we've seen. This bar in the middle here shows you from the beginning of the study to the end, which ways the antibody went. If they go over here, it's bad because they get worse. If they stay the same, it's not good. But see, for 20 patients, for both what we call class 1 and class 2, very significant reductions in the antibodies that allow us to transplant 20 of 20 patients, and despite these very high levels of antibodies. And I'll show this later, we'll talk more, but one of the important things we saw looking in the blood of these patients at about 6 months post-transplant, we found the increase in what we call regulatory T cells and regulatory B cells. These are sort of like the cops that turn off inflammation. And, you know, when you have rejection, you have unmitigated inflammation in the kidney. So having these types of cells in your body or being able to induce them can result in a powerful anti-inflammatory and immune regulatory response that hopefully would reduce the need for other drugs. And one of the, if we go, moving on to regulatory T cells, just a few words about them. This is nature's immune regulators. These are cells that turn off inflammation in the human body, and defects in these cell lines often result in autoimmune diseases. And if we can restore those regulatory T cells, we can get rid of autoimmune diseases. And the same is true for transplantation. Many animal models have shown that regulatory T cells can prevent rejection. And so this is without using standard immunosuppression, which just suppresses all types of T cells. So what we want to do is to immune modulate, that is, increase the regulatory cells, get away with the inflammatory cells, and then hopefully that we can keep the graft stable without too much medicine. Now, there are two ways to do this. And we're looking at both, but we'll talk about that in a moment. But we have the ability to take blood from a donor and recipient, or mix it with the recipient's blood. And with this sort of mixed lymphocyte culture, we can generate what we call antigen-specific Tregs, so that the regulatory T cells, if we use things like interleukin-6 that you saw earlier, and that we saw earlier, we can expand these cells and then give them back to the patient to help bump up those cells in the body and prevent rejection and reduce other types of immunosuppression. So this is a bit cumbersome and very expensive, but one of the things that we have seen with IL-6, and we think that it's probably a better way to go forward, is just treating the patients with it expands those regulatory T cells in their body and would eliminate the need of some of the immunosuppressive agents. And at Cedars now, we do have some patients just on anti-IL-6 and another monthly infusion, so they don't take any pills during the day. So that, I think, will be the next phase of therapies, hopefully, for our patients. And this just shows you what the regs, these are people who receive the drug up to 6 months after transplant. You can see that we really get a big increase in those regulatory T cells. Now, I want to close by talking about another area of interest to us, and that is, if you look at, in this study, one of the studies we just finished was supported by Tom Moen and the One Legacy Foundation for organ donation, because one of the biggest problems we have today in transplantation is that we don't have enough organs. We get a lot of organ offers, and about 30 to 40% of them are just turned down because the organs are felt to be of poor quality for a number of reasons. Now, can we find a way to enhance the use of those organs and allow them to work better? And one of the things that we've been working on is a drug called C1 esterase inhibitor, which inhibits complement activation. We think the complement system, and it's a system of inflammatory proteins in the body, is very important in damaging kidneys from what we call ischemic or low oxygen that they are exposed to. So we've had a study we did, and this is the first paper we published, where we did a blinded study where we had a placebo control. And what you can see here is that for patients that received the drug intraoperatively, IV, and then one day after, that there was, the black line here, there was a benefit in reducing the numbers of dialysis. But more importantly, we saw big differences in GFR at 1 year. So their glomerular filtration rate was improved. Now, we followed this up with a study 3.5 years after that. You can see this, all these red dots here and sort of the gray dots. The dotted line are the patients who received the drug 3.5 years later, only at the time of transplant. And this line shows that you're declining kidney function down to about 30 cc's a minute from about 55. And this sustaining of kidney function is a very important thing, because these were kidneys at risk for failure. And you can see here that this line is going the wrong direction. And after 3.5 years, indeed, we did see that there was a significant difference in the graft survival in this patient group. So it looks like by treating people early on, we can improve graft function over time. And we have been very interested in this. And we developed this study, which last year I talked about, is that really a future where we would, because of the expense, we wanted to just inject the kidney itself. If we got a kidney offered by One Legacy, we would put this drug inside the kidney. We want to see if it worked, but we wanted to do it the right way. So we did a blinded study where we didn't know what the kidney was getting. So inject it into the kidney or get placebo, and then go out to about 6 months. And we're up to almost a year now, looking how the kidney functions and looking at a number of other things. So the study now is almost complete. So I wanted to show you how this looks. And I have a little video. This is Dr. Irene Kim getting ready to do a kidney transplant for a patient enrolled in this study. And I'm going to play the video. It's about a minute video. So that's the study drug that she's looking at. It'll get a little bit louder. So the pharmacy blinds us, so we don't know what we're doing, or not. We know what we're doing, but we don't know which drug is coming in, or placebo.
That's wild.
Mm-hmm.
It's wild to see that.
Yeah. Yeah. So, mm-hmm. Okay, so that's how it was done. So the surgeons didn't know what they were giving. And so the study drug was put in. The kidney stayed on the back table for about an hour or two while they prepared the area for putting the kidney in. And basically, we finished the study. And I'm going to go, this is the data that we have. And you can see out at 9 months here, very big differences in graft and the kidney function in terms of the GFR. And this was very well tolerated. So we think that this type of treatment would be more facile than giving the drug IVs to the patients. But and one of the ideas we have is it would improve, could result, or probably will result in a new perfusion solution that will help preserve this kidney function and also other organs, and might allow them to work better. So this was supported by the Tom Moen and the One Legacy Foundation, the study, primarily because we want to utilize more kidneys. And, we are taking kidneys now that other people are turning down because we know we can make them work better and give patients a good chance for a long-term graft survival. And just ending here, this was a paper or editorial that was written about our studies on imlifidase, which is the drug that cleaves the IgG in half. And this is a paper that was, our article is published in Reuters about one of our patients. And this is a young lady who had been on dialysis for about 8 or 9 years and had 100% antibodies. And we gave her this drug and were able to transplant her now 7 years ago, I think. So she's now looking to start a family and living her life. And I think that it's very gratifying to see this, that from that beginning of 1997, we didn't have anything. Now we do have a lot of things, and these will be coming along to help patients out, to get rid of antibodies and to help them receive transplants. And this is a picture that one of my friends took several years ago in Corcovado and Rio de Janeiro, Brazil. It's a very beautiful photo, I think, of the Christ on top of the mountain there, and the clouds just breaking up. But I think, just from my standpoint, that in my own career of medicine, I feel like the most important thing we can do is try to give people hope. Many times that's hard to do, and many problems are beyond the resolution of medicine. There's where we can't really solve them, but we should never give up to restore patients to health and happiness for them and their families, which is so important. So I really do feel transplantation is the best thing we can do for our patients with end-stage kidney disease, and we continue to focus on these new and evolving therapies. Fortunately, we're getting more drug companies with very novel medications and interested in kidney transplantation, and I'm hopeful that that will will allow us to do even better treatments later on, and help people get transplanted and also use more kidneys. So thanks for your attention.
Thank you, Dr. Jordan. We have a few questions for you, but
okay.
I love that hope is so important. That's my middle name pretty much.
Yeah.
But, I'm just going to tell you that you're going beyond hope, because I had a realization about a year ago that an illness is too demanding when you don't have hope, right? We need hope. But you are at the next stage after hope is flourish, and you are helping patients flourish. And, I think that is just, and I'm an example of that. So we have a few questions. The first one, I know I'm head of the Stanley.
You are a good example of that.
I am a Stanley Jordan fan club person. I think I'm the leader of it. I know there's many though.
Okay, thank you.
So somebody asked, can this approach work for nephritis?
Yes, that's a very good point. And we look at it, and I do see a lot of these drugs, moving over for treatment of glomerulonephritis. And one in particular that we've been using, I still see a lot of patients with autoimmune kidney diseases, and we use the anti-CD20 or drugs like rituximab, or a new one called Gazyva, that are, I think, the most effective therapies I've ever seen in treating glomerulonephritis, and a big advance. Many people still in the era of looking at hydrosteroids, which we don't like, or Cytoxan, which is very, toxic to the bone marrow and can cause problems with fertility and, bladder hemorrhage. So these drugs are very focused on depleting B cells that are, and they seem to be very, very important in mediating many forms of glomerulonephritis, and have been, I think, studies now have shown they're very effective for most autoimmune kidney diseases. So this is what we do, and we have been doing it actually for many, many years now, getting around the toxicity with a more focused, effective therapy. So.
Another question is, do you test every recipient candidate for IL-6 elevation?
Yeah, that's a great question. We have done that. And, and if you look at, the most important thing here to realize is that most patients on dialysis have pretty high levels of IL-6, or the surrogate for that is a protein that is induced by IL-6 called C-reactive protein. And this is sort of a poor man's IL-6, I would say. And we use that a lot, because if that's elevated, that means you have a lot of IL-6 in your blood. And unfortunately, on dialysis, we do see chronic inflammation that is mediated by IL-6. We see things like amyloidosis, or amyloid accumulation, serum amyloid A in the skin or joints. We see chronic anemia of chronic disease. This anemia is caused by a protein called hepcidin from the liver that is induced by IL-6. And when you block the IL-6, we see a lot of this go away. And what's going on now, Nori, is even better, I think, is that there are studies. One is with the drug, we're going to be doing this at Cedars with our nephrology patients on dialysis, is to treat people on dialysis with the IL-6 blockers versus placebo, look at the reduction of these inflammatory markers, and also look long-term at look at cardiovascular events, deaths from heart attack, stroke, things like that, which we think we can reduce with these treatments. So this is becoming into the kidney area as a very big potential treatment for all stages, all the way through. And so I think it will be interesting. These studies are underway now, or will soon be. And I'm very happy to see that.
Well, and we have one final question, and I think will the new protocol benefit past transplant recipients or only patients that receive the new T-cell protocol at the time of transplant?
Yeah, that's a great question. Now, this is the short answer is yes, it will potentially benefit. What we need, and if you look at the desensitization studies I showed you, those were pretty much in all retransplant patients. So we can induce those Tregs, and I think that's going to be the next phase of immune therapies. That when you get a new kidney transplant, you will maybe have a short period of tacrolimus and then be transitioned over to a drug like belatacept and an IL-6 blocker, or maybe others as well. So getting a couple of shots a month. Now, the clazakizumab, the drug that we've been investigating, that is a sub-Q 1 cc injection monthly. So having a drug like that could potentially be given at home, like an insulin shot, to prevent or treat antibody rejection, as part of your immune suppressive protocol, I think is going to be a big, big step forward. And I see that coming. I think that's what we will be seeing in the future.
Well, they always say chronic rejection has been the biggest issue in kidney transplant, and it looks like you guys are tackling it at all sides, that will help us not need another transplant, and then we'll eliminate the kidney transplant list. One final question, if you feel comfortable answering it. Somebody asked about the artificial kidney.
Yeah.
And do you have any comments on that? I mean, we have a podcast on it. But in your opinion, do you think the artificial kidney will be seen in the next 5 to 10 years?
Well, in some version, I think, the question is how that will compare to a kidney transplant. And so that's obviously, the kidney transplant does much more than just provide clearance of urea and creatinine and water balance, because, it has hormonal functions as well, making epogen and that sort of thing. So that would be the question. And I think certainly every aspect of this therapeutic intervention should be looked at, because we just don't have enough kidneys to transplant everybody at this point. And I think that and the other part of this also is this whole thing. I know people are interested in xenotransplant, in the papers about using the pig hearts, the genetically altered pigs, and that's still a bit far away from my view too. I think it's doable, but I think the requirements for immunosuppression, prevention of rejection, metabolic issues as well would be, it's going to have to be worked out pretty well before we can move forward with that type of therapy. And how much it would impact what we're doing right now in terms of the list would be of interest. It would certainly eventually could do that. So we'll just have to see, but it needs some more refinements, I think.
Well, what I think the holy grail is that if you can keep people off dialysis with the new drugs, and then, move it forward to keep the transplants that are existing. That's the low hanging fruit.
It is, you bet.
It's the
Yeah.
low hanging fruit.
Yeah, and what I do, when I see people, I go to my clinic and I come in, I have my GN patients and I say, "Oh my God, you say kidney transplant." I said, "No, no, no. This is my job is to keep you off the kidney transplant list and make your own kidneys work longer." So that's what I like to do. If we could do that for
That's the
everybody.
ultimate. Well,
That's the ultimate, yeah.
we're on behalf of patients. Thank you so much for your dedication. I just put
Thank
in
you.
the chat, RSN just launched a transplant dashboard to make it easier for people to find the different centers, because we actually had a great story. One of our living donors works for a company called Salesforce, and they volunteer hundreds of hours to make the data easy for patients to understand. And I just put that in the link. It's going to go live. We're going to send it out next week. But I also wanted to make one quick point, and thank you, Dr. Jordan. You are absolutely amazing. I wanted to make one other quick point that about 40% of the list is inactive. And it's our job as patients to make sure that we're pushing that, making sure that we know if we're on the list, if there's a test we need to figure out, something like that, that we are able to advocate for ourselves, so that a friend of mine needed a test and it took her like, "Oh, we can't see you for 2 months." She called every day till they got a cancellation. And so she could get active back on the list, because it's mind-boggling that 40% of patients are not active right now. And that was one of our goals with the transplant dashboard, is to educate people about this, and then also about the different centers. Like Dr. Jordan has a very large center. So we had questions earlier on about, if one center doesn't give you what you want, in the sense of, they're, "Oh, we don't do that. We don't do, that kind of transplant," or whatever. Another center may do the transplant. So they're not all the same. It's a medical practice, everybody. And a shout out to our corporate mission partners for making this happen.